Question number 89, non-Hodgkin's lymphoma. For this question, before we really get into the depths of non-Hodgkin's lymphoma, I'm really quickly going to recap the differences between a lymphoma and a leukemia. And I'm going to also cover some of the similarities and differences that exist between a Hodgkin's lymphoma and a non-Hodgkin's lymphoma. And then, of course, we're going to go into the depth about non-Hodgkin's lymphoma. The whole idea is I give you a holistic and overall picture of everything that you could be asked regarding this question instead of just focusing solely on non-Hodgkin's lymphoma. Obviously, the leukemia questions and the Hodgkin's lymphoma questions are separate and you're going to have to learn them anyways. But for a whole holistic video, I am going to cover the bits and pieces that are required for the other two aspects and then talk about non-Hodgkin's lymphoma. Okay, so lymphoma. A lymphoma is simply a solid cohesive neoplasm of the immune system cells. Solid cohesive neoplasm of the immune system. Right, and essentially what you've got is your hemopoietic precursor cell. I'm going to abbreviate it HPPC, which undergoes a malignant proliferation. And what basically happens is, in the case of a lymphoma as compared to a leukemia, this malignant HPPC or hemopoietic precursor cell proliferates and produces discrete masses. And so the important thing to focus on here is the production of discrete masses. These are well defined and clear-cut masses that are produced in the bone marrow and also produced in any site that the cancer metastasizes to. And so this is essentially what a lymphoma is. Compare this to a leukemia, and what you have is that the malignant cells diffusely involve the entire bone marrow and will eventually spill into the circulation. And so your basic differentiator between a lymphoma and a leukemia is that a lymphoma is going to form cohesive discrete masses, whereas a leukemia is going to follow the same exact steps, but instead of forming discrete masses, you're going to have diffuse cells that are more likely and in fact will spill into the circulation in higher numbers. And so an important thing to note with lymphomas is that they usually do not spill into the blood at high numbers, and they are always lymphocytic cells. Leukemias, as you would know from the previous questions, can be, just going to put it up here, leukemias have diffuse cells that spill into the bloodstream in high numbers, and they can be both myeloid cells or lymphoid cells, whereas lymphoma is always only lymphocytic in nature. Right, and so that is just a basic overview of the difference between a leukemia and a lymphoma. Now we need to discuss some differences that are pertinent to us in understanding non-Hodgkin's lymphoma and comparing with Hodgkin's lymphoma. There's obviously a whole other question on Hodgkin's lymphoma, which will cover everything in a bit more detail. But for now, just for basic understanding, we're going to talk about some of the differences and some of the similarities as well between Hodgkin's and non-Hodgkin's lymphoma. So, first thing we need to talk about is the clinical presentation of Hodgkin's lymphoma and compare it with non-Hodgkin's lymphoma. And as far as we need to know, Hodgkin's lymphoma usually arises in lymph nodes that are relatively easily accessible. There are, of course, exceptions, but for the most part, this arises in lymph nodes rather that are relatively easily accessible. What do I mean by this? So these are more of your superficial lymph nodes that you can easily, palpate, you can easily see. Examples would be your supraclavicular or your occipital, so on and so forth. Right? Compare this to non-Hodgkin's lymphoma. The lymph nodes that these cancers arrive in are relatively inaccessible, and in about 30% of the cases, the origin of non-Hodgkin's lymphoma is from extranodal tissue. So they do not necessarily have to begin from lymph nodes. And so that is the first differentiator between Hodgkin's lymphoma and non-Hodgkin's lymphoma. Second thing is to do with how these cancer cells or these metastatic cells, malignant cells, how do they spread? And the spread of Hodgkin's lymphoma is usually said to be in an orderly fashion. And the word you'll see thrown around is contiguous spread. What this basically means is that if these cancer cells or metastatic cells are found in one group of lymph nodes, they are going to spread to lymph nodes that are next to each other or next in line. It's not going to be as if you have these cancer cells present in the supraclavicular lymph nodes and then they end up somewhere around the abdominal lymph nodes. That's not how it works. So you have contiguous spread, which goes from one lymph node to the next lymph node to the next lymph node in an orderly fashion. Compare this to non-Hodgkin's lymphoma, and you can already imagine that their spread is disordered. There is no order in how these cancer cells will spread, and so it has disordered spread, which is completely unpredictable. And so you can imagine this normally makes it a lot harder to understand the prognosis and the further not complications, but rather the further treatments that are involved with non-Hodgkin's lymphoma, because its course is relatively very unpredictable. Right? Now this is another important differentiator. In the cases of Hodgkin's lymphoma, you will see systemic manifestations of inflammation. Whereas in the case of non-Hodgkin's lymphoma, this is relatively rare or usually not seen. Because of how these cancers and each of these lymphomas spread, non-Hodgkin's lymphoma has a predictable response to treatment, whereas because of the diversity that exists in non-Hodgkin's lymphoma, the response to treatment is unpredictable. Now, a key differentiator in terms of histopathology is the presence and the relative absence in non-Hodgkin's lymphoma of Reed-Sternberg cells. And so Reed-Sternberg cells under the microscope usually have two nuclei, prominent nucleoli, have that owl-eye appearance, which you will remember from some of the microbiology videos, and they can also have bilobed nuclei. In the case of non-Hodgkin's lymphoma, these are simply not present. So Reed-Sternberg cells are absent. And one last differentiator is that Hodgkin's lymphoma normally has an efferent lymph node spread, whereas non-Hodgkin's lymphoma can also have hematogenous spread. And when we get to the extranodal manifestations, we'll talk about this in a bit more detail. And so these are some of the important things that you need to be able to talk about in terms of differentiating non-Hodgkin's lymphoma from Hodgkin's lymphoma. And one other important point which I will also mention is that Hodgkin's lymphoma has something we mention as bimodal distribution in terms of its prevalence. And so if you graph it out, it look something like this. Whereas the chances of someone developing a non-Hodgkin's lymphoma just increases with age. And so key points to talk about when you're differentiating these two is its cellular composition, the epidemiology, the manner of spread, whether or not systemic signs of inflammation are present, and what the prognosis is for each of them. And that should be it as far as differentiating these two go. Now we're going to talk about non-Hodgkin's lymphoma in a lot more depth, and we're going to talk about the different types of phenotypes associated with non-Hodgkin's lymphoma. So with non-Hodgkin's lymphomas, there are 4 parameters according to which clinical pathological diagnostics of the lymphoma are done. This includes the genotype, in which we're going to talk about the relevant chromosomal abnormality that takes place. We're going to talk about the phenotype, which basically means we're going to discuss the type of cell involved. We're going to talk about the morphology, in which case we discuss the distribution of the malignant cells. Example, whether they're diffuse, whether they're follicular. I'll explain what these terms mean in just a bit. And then, of course, we're going to have to mention some clinical features as well. Right, so phenotype, we'll just talk about phenotype really quickly, just so that's out of the way. When we talk about phenotype of non-Hodgkin lymphoma, it just basically means we're trying to describe the cell origin, the type of cell, the predominant cell that exists, that has undergone this malignant transformation and is responsible for the non-Hodgkin's lymphoma. And from the diagnostic point of view, diagnosis, if you guys really want to know, is done through flow cytometry, and obviously biopsies and all work too. But that's normally not done in the cases of EBV infections. More on that in a bit. But what you really want to do is you need to be able to tell which CD surface antigens are present on T cells, NK cells, and B cells. And this is how diagnosis is actually done through flow cytometry. I'm not going to go through the mechanism of how flow cytometry works, but all you need to know is that it uses the antigens that are present on the surface of these T cells and B cells and NK cells, the CD cluster differentiation proteins that are present, to differentiate between it. So T cells have CD 2, 3, 4, 7, 8. NK cells have CD 56 and 16, whereas B cells have 0, 19, 20, 21, 22. Excuse that 0, wasn't supposed to be there, my bad. So that is a bit about non-Hodgkin's lymphoma. Now, before we get into the depths of the different types, we really need to discuss what the lymphocytic pathway is. What happens to a lymphocyte when it's produced by the hematopoietic precursor cell, where it goes to, how it matures, because that will really help in understanding the different types of lymphomas that can arise. And so how it begins is that you have your hematopoietic precursor cell in the bone marrow, which undergoes its first line of differentiation and forms a lymphoblast. Now there are two places where maturation of lymphocytes take place, one of them being the thymus and the other one being the lymph nodes. But before we get to that, this lymphoblast now will enter the blood circulation, and it can either go to the thymus, where it undergoes certain processes and can either become a CD4 plus T helper cell or a CD8 plus T cytotoxic T killer cell, right? And as far as the lymphomas are concerned with respect to T cells, they're relatively rare in nature. They only account lymphomas of these cells account for only about 10 to 15% of all of the non-Hodgkin's lymphomas. So you can have something that is a T cell non-Hodgkin's lymphoma. B cells, however, account for about 85 to 90%. So that's going to be more what we're going to be discussing in this video in general. But just know that T cell non-Hodgkin's lymphomas do exist, and they are normally seen due to some mutations occurring because of an infection by the human T lymphotropic virus 1. So this has a very high association with T cell non-Hodgkin lymphomas. For now, this should be enough really. Moving on to B cell lymphomas now. Once in the blood circulation, this lymphoblast will further mature into what we call a naive B cell. And now this naive B cell will enter the lymph node. First zone it enters of the lymph node is what we call the mantle zone. And from immunology, if you remember, at the level of the mantle zone is where these naive B cells are first exposed to antigens. So they do have receptors present and they're going to be exposed to antigens, right? Once this exposure to antigen does occur, we have the process of somatic hypermutation take place. Just going to describe that in a bit. But another important thing to notice before I get into that is that if this naive B cell is not treated with an antigen exposure, it's not exposed to any antigen at all, it remains arrested in this mantle zone phase. So I'm just going to draw out a mantle zone B cell which has not been exposed to any antigen, and it remains in this naive state within the mantle zone itself, right? Now the process of somatic hypermutation, just a bit in general to talk about if you do not remember it from immunology or biochemistry. What basically happens is once your cell has been exposed to a particular antigen, it will undergo a programmed process of mutations of the variable regions that exist within the immunoglobulin genes. And the basic idea of this is to help in the B cell and its receptors increase its affinity towards the specific antigen, and to be able to mount an immune response to almost any antigen that it can then be exposed to. So that's literally all you need to really know about somatic hypermutation with respect to pathology. Nothing too serious, but what you need to know post that is that once this antigen exposure has taken place, you are going to have a follicular blast cell, which upon further antigen exposure will now form what we call a centroblast. And now centroblasts have many receptors on them which are primed and have been exposed to antigens. And so before these centroblasts can now further differentiate into centrocytes, what needs to happen is that you need to have some sort of mechanism to check whether or not the immunoglobulins, once the somatic hypermutation has taken place, once antigen exposure has taken place, that these cells, that these receptors that are present actually have a good affinity, a high affinity for antigens. And so if they have a low affinity, they undergo apoptosis. End of discussion. They're gone. Bye-bye. Never seen again. However, if they do have a high affinity for the antigens that they were primed with, at this stage, they can undergo further maturation into a centrocyte. I do not have space to draw it out, so what I will do is just mention that these centroblasts now will become centrocytes, right? And now through further stimulation by T cells and dendritic cells, these centrocytes will now undergo a process of class switching. And that, if you remember again from biochemistry, class switching basically means once you change one isotype of your immunoglobulin to another type. So they can have IgG, it begins as IgM rather, forms IgG, can form IgA, so on and so forth. Not really too relevant, but just something you need to understand in general, right? Once all of this happens, once all of this is done, you have a marginal zone lymphocyte. And so these marginal zone lymphocytes are the ones that can now differentiate into B cells that are memory cells, memory B cells, or ones that are your plasma B cells that produce antibodies. And so the basic point of telling you this lymphocytic pathway is that mutations can take place in different parts as these processes are going through within the lymphocytic pathway. And as a result, you can have different types of lymphomas, the different phenotypes that exist of, the different morphologies and the phenotype that exists of a non-Hodgkin's lymphoma can occur depending on where mutations take place along this lymphocytic pathway. And so the important ones that are to be mentioned are at the mantle zone, are at the level of the centroblast and centrocytes and the follicular blast cells, and at the level of the marginal zone lymphocytes. And so, as mentioned here, the lymphomas can originate at any of the above stages, but the major ones that we're concerned about are the ones I put an asterisk next to. And the complications of this can be immunosuppression and autoimmune problems. More on the complications a little later. So let's talk about some of the individual phenotypes that are involved in non-Hodgkin's lymphoma. And so the first one is small lymphocytic lymphoma, or in some cases you'll see that this is also classified together with chronic lymphocytic leukemia. And this is simply, again, do not confuse leukemia and lymphoma. The only reason you'll see in some resources that these two different types of cancers are grouped together is because they have this, at the histological level, they have the same types of cells making up the malignancy that we're talking about. The only difference is one of them ends up forming a lymphoma, which is a discrete mass. The other ends up forming a leukemia, which has diffuse cells spread throughout the bloodstream. The whole point is, though, that the underlying cell, that the bottom line is that the cell that makes up these two malignancies are literally the same. And so small lymphocytic lymphoma, our focus here being on small lymphocytes, basically means that it gives away that these are relatively less aggressive, they're less transformed, and are less malignant. Some important things you need to know about this is that this is most commonly seen in the elderly population. This is indeed a B cell lymphoma, so B cells, but what you will need to know is that normally small lymphocytic lymphoma presents with one T cell marker, which is normally not seen in B cells, and that is CD5. Some other important features are the fact that the bone marrow is almost always involved. And so a complication of this is pancytopenia. And because of pancytopenia, you're going to have immunosuppression because you do not have functioning white blood cells that can fight off any infection. The other thing is that if you have immunosuppression. Immunosuppression in this case is basically just characterized by the fact that you cannot produce enough antibodies, or the patient is in a state of hypogammaglobulinemia. What are some morphological changes that you need to know? First thing is that small lymphocytic lymphoma, as you'll see, is probably consistent with most of these lymphomas anyways, is that it infiltrates the liver and the spleen, and so you can imagine easily this causes hepatosplenomegaly, and it has infiltration to the lymph nodes as well, and this can cause generalized lymphadenopathy. Chromosomal abnormalities that are associated with this are usually trisomy 12 or deletion in chromosomes 11 and 13, right? One other morphological point of mention is that this has a diffuse pattern. Now, if you look under the microscope, obviously you do not need to know this, but for your information, a common finding that is seen with this is that the cancer cells that we're talking about can end up actually rupturing in your histopathological assessment. And so this has a classic presentation under the microscope of something we call smudge cells, just for your information. And just one other thing you need to know before we move on is that, let's say you have one of these small lymphocytic lymphoma cells, one of these cells and one of these malignant cells ends up going into a lymph node. It's very much possible that they can convert into a diffuse large B-cell lymphoma. We'll talk about this in following on, but this process where you have one malignant cell converting into a diffuse large B-cell lymphoma once it enters a lymph node is known as Reiter syndrome. I hope that's how it's pronounced, but either way. Now, coming to the second type, follicular lymphoma. Name gives away. This lymphoma consists of cells that are arranged in well-defined follicles. As far as characteristics go, these cells are relatively differentiated, and the way it's quite easy to remember whether or not a cell is well differentiated or not as well differentiated right from its name in this case is you can go back to the lymphocytic pathway. You can see here that we've got follicular blast cells. They have relatively better differentiation than a lymphoma that would arise consisting of cells that are from the mantle zone, for example. And so that's one way to know whether or not how well a cell is differentiated with respect to its name. It can be a giveaway if you want to remember it that way. Now, because these are relatively differentiated, they normally present with an indolent course and are seen most commonly in the elderly. At the histological level under the microscope, you'd want to know that follicular lymphoma consists of aggregates of centroblasts and centrocytes. So these are the two cell types that are more commonly seen, centroblast and centrocytes. And once again, going back to the fact that by the name of the type of cell that is present, you can have an idea of whether or not the entire of the cancer cells are well differentiated or poorly differentiated. So again, you remember from the lymphocytic pathway that centroblasts and centrocytes are one of the rather well differentiated phenotypes of these cells in its development process. Now, coming to chromosomal abnormalities that are associated with follicular lymphoma, is the translocation of the BCL2 gene, translocation BCL2 to the loci of the immunoglobulin gene. The immunoglobulin gene is found on chromosome 14. The BCL2 gene is found on chromosome 18. So it's an 18-14 translocation taking place. BCL2, the IG gene loci. And once this happens, you form a fusion protein that results in the prevention of apoptosis. And you'd remember why apoptosis was an important process taking place within the centroblast and centrocyte cell types, because you need the cell to be able to recognize a antigen and have a good or high enough affinity for these antigens. If these cells do not have a good affinity towards a specific antigen that they are supposed to be primed for, they are killed off through apoptosis. So what this BCL2 IG gene translocation does is that it produces a fusion protein that prevents apoptosis. So one question you may be asked is how you differentiate between a reactive secondary follicle that forms, and how do you differentiate this oh my god, I'm so sorry. How do you differentiate a reactive secondary follicle from a follicular lymphoma? You may be asked this question in the exam, and the giveaway is simply that in the case of follicular lymphoma, you have low amounts of mitotic figures and no apoptotic bodies. Versus in the case of a secondary reactive follicle, which will have high amounts of mitotic figures and apoptotic bodies. What about the presentation of follicular lymphoma? It normally presents with painless enlargement of the lymph nodes, but the most important thing you need to know with respect to follicular lymphoma and its prognosis is that A, it normally has a poor response to chemotherapy, and probably the most important thing that you really need to remember is that follicular lymphoma can progress to diffuse large B-cell lymphoma. Again, we'll talk about that in just a bit. Now, coming to mantle cell lymphoma, the third type that we're going to discuss. This is a relatively quick one. This is to a B-cell lymphoma, and like we discussed with small lymphocytic lymphoma, these B cells also present with the T cell marker CD5, and so that's something that helps in diagnosis. Now, coming to the chromosomal abnormality that is associated with this, it is the translocation of cyclin D1 from chromosome 11 to chromosome 14. Once again, all the translocations normally take place to chromosome 14, which is where the IG gene loci is. Loci, however you want to pronounce it. And the basic idea is at this loci, you have high amounts of proliferation taking place because you need high amounts of immunoglobulins being produced as part of the immune response. And so the basic idea now is that this cyclin D1 gene is now translocated to a loci where there is a higher amount of expression in general and higher amounts of translation taking place. And as such, you have a fusion protein produced, as you did with follicular lymphoma. So this fusion protein, as compared to follicular lymphoma, This fusion protein increases the rate of proliferation of the cells. And what type of cells specifically? Cells that are found in the mantle zone. So mantle cell lymphoma. You go back to the lymphocytic pathway and you will see that the mantle cell type was one of the first cell types that we encountered within the lymph nodes, and these were cells that were relatively poorly differentiated. Go back to how pathology works, go back to how tumors work. If you have a cell that is relatively poorly differentiated, you'd imagine it has a higher chances of being a malignant and higher chances of being a cancer that has relatively poor prognosis and a cancer that is relatively aggressive. And so what you need to remember is that mantle cell lymphoma are aggressive neoplasms and that they normally follow a diffuse pattern. Other presentations include painless and generalized lymphadenopathy. And another important presentation is that of lymphomatoid polyposis. And what this basically is a distinctive and particularly rare clinical type of malignant GI lymphoma. And so mantle cell lymphoma, one of its presentations is lymphomatoid polyposis, which is a malignant GI lymphoma. Cool. That should be pretty much everything you need for mantle cell lymphoma. Now coming on to the big fish, diffuse large B-cell lymphoma. Mentioned it quite a few times already, but let's get into it. So diffuse large B-cell lymphoma is the most common non-Hodgkin lymphoma, and you need to remember this. The name gives away large B cells, so it has large cells, and the name also gives away that it is diffuse in nature. And so you can have an inference from these two features alone that this is a highly aggressive neoplasm having a highly aggressive clinical presentation and behavior. And so histopathologically, looking under the microscope, how does a large cell look like? Obviously this is a very rough scheme. If you look under a microscope, it'll be a little bit harder to differentiate, but having said that, a large cell may consist of the following features that you probably should know. The nuclei may be poorly rounded, or relatively well rounded rather, but they can be regular or irregular as well. They have multiple nucleoli, and they may look like centroblast as well. And I'm guessing now you can have an idea of why they may look like centroblast, because we did mention that follicular lymphomas, in which the centroblasts and centrocytes were the most common type of cells found within follicles, could progress to diffuse large B-cell lymphomas. And so that will be the first chromosomal abnormality that diffuse large B-cell lymphomas may be associated with. To be precise, about 30% of diffuse large B-cell lymphomas show the same chromosome 18 to 14 translocation. And this is simply because this B-cell lymphoma was a progression from follicular lymphoma. Right? The second type is associated with a mutation in the BCL6 gene, which is found on chromosome 3, and this is a sporadic mutation which results in the overexpression of the BCL6 protein. This is seen in about 30% of the cases as well, and the remaining 40% are overall just sporadic mutations that increase the likelihood of someone developing diffuse large B-cell lymphoma. Right now, clinical presentations and how these lymphomas actually come about, other ways that they can come about, their presentations. It's gonna go in red. First one is an Epstein-Barr virus driven stimulation of B cell, which results in their proliferation. And this is normally seen, if you remember from the infection of EBV patients, this is normally seen in the immunocompromised, so those who are AIDS patients or are immunosuppressed. And it's important to note that partial regression could take place, not necessarily has to, but could take place if the state of immunosuppression is reversed. And obviously immunosuppression can also be caused by drugs. It doesn't necessarily have to be someone with HIV, with an HIV infection. And so partial regression may be possible if you have removal of any of these immunosuppression drugs. Another cause of diffuse large B-cell lymphoma, which you'll remember from microbio, is infection by HHV8 virus, which is associated with a homologous cyclin D1 protein that stimulates the over proliferation of infected B cells. And clinically, these are the ones that spread to cavities. This could be your pleural cavity, could be the pericardial cavity, it could be the peritoneum, and this can lead to effusions within these cavities. The type in which diffuse large B-cell lymphomas may present is they can present as mediastinal masses. These are normally seen in females, more commonly seen in females rather, and they can spread to the abdominal cavity and to the central nervous system. And so that is diffuse large B-cell lymphoma. Last two types we need to discuss are Burkitt's lymphoma and extranodal marginal zone lymphomas. So Burkitt's lymphoma, you will remember that there were, from microbiology, that this was associated with EBV infection, Epstein-Barr virus infection. And there are two types, the endemic African presentation and the second being sporadic. Now, the endemic variant almost has 100% Epstein-Barr virus involvement, and these are normally seen in the jaw and the maxilla, these lymphomas, Burkitt's lymphomas, whereas sporadic normally have a 20% EBV association, and they are more commonly seen in the abdominal cavity. Important things to note about Burkitt's lymphoma and the two different types of Burkitt lymphoma is that they are histopathologically similar. So histopathologically similar. However, their clinical and biological behaviors are different. They have an aggressive onset and progression, and they normally consist of cells that are derived from the germinal center, Cells in the germinal center as per our lymphocytic pathway diagram included follicular blast cells, centroblasts, and centrocytes. You look under the microscope, what you're going to see is something we call the starry sky appearance. What this basically consists of is that you have rapidly proliferating cells that are usually larger than normal cells. So as you can see here, these are larger than normal cells and they are loaded with chromatin, and so they're relatively darker. In fact, it's this bit that is like that. So these cells. Whereas you can also find the presence of apoptotic bodies taking place. And then once you have apoptotic bodies present under the microscope, you will see that there will be an infiltration by non-neoplastic, and this is important, non-neoplastic macrophages that are here to digest these apoptotic bodies. And so that is what forms the star bit of the starry sky appearance. Burkitt's lymphoma. What you need to know as far as chromosomal abnormalities go is that this is associated with a translocation from chromosome 8 of the MYC gene to the same chromosome 14 IG gene loci. And that's about it for Burkitt's lymphoma. Coming on to the last one we didn't need to talk about, extranodal marginal zone lymphomas. Again, you'll remember marginal zone as the one with cells that are relatively well differentiated. And so these are by nature low-grade B cell tumors. And these are more often seen developing in malt tissue. Malt is mucosa-associated lymphoid tissue. And so these are the ones, these neoplasms are normally seen in the parotid gland, which can occur secondary to Sjogren's syndrome, which you'll remember from, or rather you will do moving forward in GI pathology, so not too much on it now, but just the fact that this is a chronic autoimmune inflammatory process that results in an increased number of lymphocytic an increased number of lymphocyte stimulation and infiltration in the salivary glands, in this case the parotid. And then the other place where you have mucosa associated lymphoid tissue is in the GIT. So GIT is another common site where extranodal marginal zone lymphoma can occur. And this normally, within the GIT, this normally occurs secondary to an H. pylori infection. Again, more on that when you get to GIT pathology. But what you need to know for now, which I want you to remember when you do GI patho as well, is that a maltoma or a a mucosa associated lymphoid tissue, lymphomas arising from these are called maltomas as well, and that a maltoma that is caused by H. pylori usually will regress if the bacteria is eliminated, as the T cells that are stimulated by H. pylori and secrete growth factors that support this maltoma growth are then removed. And so that is pretty much it as far as the issue goes. And what another important thing that you really need to know is that extranodal marginal zone lymphomas can also develop in non-mucosal sites, and these would normally be the orbits and the breasts. No real need to talk about anything else. In this, the last thing to cover is your chromosomal translocations. in this case, and this is simply the translocation of the BCL10 protein gene, which is found on chromosome 1, to once again chromosome 14, where you have the IG gene loci on chromosome 14. Right, and the last thing we're going to cover really quickly, because it's already been 15 minutes to this, I believe, is the extranodal involvement, an overview of the extranodal involvement of non-Hodgkin's lymphoma, very quickly, very simply. And so you can, number one, if you have involvement of the CNS, you can have a primary CNS lymphoma. This is normally seen more commonly in HIV positive patients, and the most common subtype of non-Hodgkin lymphoma seen is diffuse large B-cell lymphoma. Second, as far as extranodal involvement goes, as we said, you can have bone marrow involvement, which results in pancytopenia. And so a complication of pancytopenia is that the patient is immunosuppressed, and this is a common cause of death in these patients. Right? We talked about hepatosplenomegaly and lymphadenopathy. We talked about GI involvement in the case of the two different types of phenotypes that are associated with the GIT were marginal and mantle cell lymphomas. And the fifth type, which we did not discuss, but it is important for you to know because it is more associated with T cells, is a condition called mycosis fungoides. And what's basically happening in this is that you have T cells, the neoplastic T cells, that deposit under the skin, and they end up causing erythroderma, which is under the skin. They cause erythroderma, which are these red rashes that look like as if they are from a fungal infection, thus the name mycosis fungoides. And what can happen is that these T cells, they can enter the bloodstream as well, and then you can have a disseminated rash that is formed in the body. And once these T cells are within the bloodstream, so I'm just going to put bloodstream here, once they disseminate into the bloodstream, this results in this disease process then is now known as Sézary syndrome. So T cells depositing neoplastic T cells as part of a T cell non-Hodgkin lymphoma that deposits into skin tissue causes mycosis fungoides. And then once these T cells then end up entering the bloodstream and disseminate to other different parts of the body, they can form further erythroderma throughout the whole body. You can have a disseminated rash, and once this happens, this is known as Sézary syndrome. And that should be it for non-Hodgkin's lymphoma.